Hi, everybody. Welcome to the first installment of our series of pre-conference video conversations, MM&M Transform Talks. The intent here is to start the conversation on the future of healthcare marketing ahead of the virtual MM&M Transform Conference, which is slated to take place September 30th and October 1st. I'm Marc Iskowitz, Executive Editor at MM&M, and I'll be your host for this 25-minute conversation. And joining me for this conversation is my special guest, Dr. Neville Sanjana, who is core faculty member, New York Genome Center, and assistant professor of biology at New York University and of neuroscience and physiology at the NYU School of Medicine. Hi, Dr. Sanjana. It's a pleasure to talk with you again.
Hi, Mark. Thanks so much for having me. I'm delighted to be here.
Of course. I think the last time we spoke was back in May, and we talked about CRISPR and its impact on diagnostics. Today, we're going to talk about something completely different, but perhaps where you're drawing on a similar skill set, and that's your lab's work on coronavirus mutations, a topic of increasing concern these days, but with case counts due to the pandemic taking an unsettling turn. You posted on Twitter back in June, and your lab, the Sanjana lab, as recently as this month, updated the findings on the function of the SARS-CoV-2 spike mutation, otherwise known as D614G, on a preprint server. Why is this concerning? Tell me about this.
Yeah, so thanks again for having me here. So I mean, our lab, like most labs, in March, we shut down and we started working from home. And then a couple of weeks into the pandemic, we started to think, we're molecular biologists, we're geneticists, how can we contribute to some of the response? The world definitely needed help with dealing with the coronavirus pandemic. And one thing that we noticed that we thought was very interesting when we were just sitting at home is we were looking at different mutations that were occurring in the virus from different patients that had been sequenced, where their nasal swabs had actually been sequenced. And what we observed is there was a mutation that came about in early February that started to sweep through the population. It started to grow in prevalence. And here, sitting here in July, we now know that that mutation is now almost 99% of all coronavirus worldwide. But it started in February as a very small proportion, but we could already see a very sharp increase. Other data that we had seen around the same time showed that, and this was in maybe early April, showed that countries that had more of this mutation tended to have a higher fatality rate. The case fatality rate was higher in those countries where there was more of that D614G mutation in the spike protein. And I should just mention the spike protein is really what gives coronavirus its name. It is the outer covering, the little spikes you see on the viruses. So for these reasons, we thought, hey, maybe this mutation that's growing in prevalence is actually more severe, killing more patients. This really merits some further study. Around the same time, a few other groups had started to look at the viral load in patients. And two different groups, one in Washington and one in the UK, had found that patients that have this mutation in their coronavirus samples tended to have a higher viral load. You might have heard of this qPCR-based test that's actually used to test for coronavirus, whether you have coronavirus. This is the gold standard test. And what was really interesting is over a couple hundred patients, both of these groups had shown, whether the patients were coming from the United States or the UK, that if you had this mutation, the virus was detected with a much higher, the viral load was higher in those folks, meaning it was detected sooner in that qPCR-based assay. And so for all these reasons, we started to wonder, well, is the virus more deadly? Is the virus more infectious? Again, that higher viral load. And is this something that we could really dissect in the lab? And so that's what got us interested and motivated a few of the folks in my lab to really get back into the lab and really test the two forms of the spike protein, the original form, the first sequenced form that had come out of China, and the one that had mutated in February to have the spike mutation, to test them head-to-head in the lab.
Sure. So we'll talk about whether the mutation is leading to not only an increase in infectivity, but also an increase in mortality in a second. I just wanted to just kind of have you say a word about pandemic science. You put this paper out on a preprint server called BioRxiv, which I assume is a sister server to MedRxiv, which I've seen some other mentions of in the media for other kind of coronavirus-related science. But and this was picked up, by some major media outlets like the BBC, for instance, where I caught your interview not long ago. Talk about that for a second, how the urgency of the pandemic has changed the way science is being done.
Yeah, I think over the last several years, pre-pandemic, there's been an emphasis on trying to share science faster. And a lot of folks have realized that journals, though they provide some great things like peer review, the science gets out in kind of a slower process. Of course, when you're dealing with a pandemic, there really isn't so much time. You don't have the luxury of waiting. Sometimes a paper can take a year or more than a year under review. And yes, peer review is very, very important, but for things like the COVID-19 pandemic, it's been very important to get data out there, to get tests, diagnostics, information about the virus out to the widest group of scientists who themselves can act as peer reviewers for sure. It still is important to have that peer review, but to communicate it as fast as possible. And one thing that's been really great about these preprint servers like bioRxiv and medRxiv is that they allow you to upload a paper and it to be publicly available, within 24 hours, something like that. And I think that's been really important for speeding science along. Certainly one thing that's been really helpful for the work that we've done on the spike mutation, we weren't, when we initially saw these results, which we found in the lab, when we saw that the mutation actually increases the virus's ability to infect many different human cell types like lung, liver, colon, we actually were unsure about whether, is this a real effect? And we started really testing many other things. But something that gave us a lot of confidence in the first days and weeks after our paper came out was that not just us, but several other groups, all within the span of maybe a week or two, had papers out on bioRxiv. And in this sense, it wasn't really a competitive situation, but something more that gave us a lot of confidence that what we were seeing was a real effect, because other people who we didn't know halfway across the world also replicated the same science, and let us really push forward and say, okay, now that we know that this is more infectious, what might be the reasons? How do we counter it? What are the effects on vaccines? All the kind of natural next questions, but it accelerated that process basically.
Right, so seeing other labs around the world kind of address the same hypothesis and be curious about the same things is a way that kind of piques your interest, I'm sure, and is something that's good to know. And that whole process is being accelerated. The other thing that I thought was really interesting was the way your lab, which your lab is comprised of microbiologists and genome engineers. You work a lot with CRISPR, which, as I mentioned, we talked about before. But you really altered course to kind of press your people, if you will, into service to do pandemic research, which I think is very cool. So we'll talk more about that. I wanted to address the point about the infectivity and the relationship to mortality. So all viruses mutate. Obviously, this is a little bit worrisome given the infectivity rate. I mentioned that you posted it on Twitter back in June, and then you updated in July. What did you notice in that interim time that you felt the need to update the paper in July?
Yeah, I think that's another great thing about these preprint servers is there isn't any sense of a final copy of the paper, which is often what you have with journals. Here, you're able to update the science as you get new results. And so some work that didn't make it into the first version of the paper, but we were able to add about a month later, was one of the hypotheses that we had was that maybe this mutation, the change that it induces in the spike protein, we already knew that it makes coronavirus more infectious, but one question we had was how. And in the first version of the paper, we kind of explored one dimension of that. But I think one of the most straightforward explanations that you might have is that we know that this spike protein binds a receptor on human cells called ACE2. And so one thing we wondered is, okay, does this mutation, maybe it just makes spike better at binding the ACE2 receptor? And so with some collaborators here at NYU, we were able to very explicitly test that. We actually purified ACE2 protein and purified either the regular spike or the spike with the mutation. And we looked at that interaction of the ACE2 receptor and the two different forms of spike. And what we found was actually there's no difference in the binding. So that said that, this hypothesis, as nice and as clean and as neat as it sounds, actually isn't the right track. It's something else about the spike protein that's changing, that's enabling this virus to be more infectious.
Mm-hmm. Any theories on that, how the change in the spike protein leads to the increase in infectivity?
Yeah, one thing that we've seen is that in the cells that produce the virus, because of course the virus infects human cells and then kind of hijack that particular cell to make it a factory to produce more and more virions that then go out and infect other cells. So in those producer cells, one thing we noticed is that the spike protein actually has certain sites on the protein, on the spike, that are kind of pre-cut. You can think of it as like sometimes when you get a package in the mail, it already has like kind of a prefabricated cutout line that makes it easy to tear open. So spike has some sites that are kind of like that make it easier for spike to attach onto a target cell and infect that cell. And we think there's a difference in that kind of perforation, that pre-cutting process of the spike protein that's making it more and more infectious. But this is actually an area that we're still working on.
Okay. Now, you mentioned that you've seen this in a lot of different types of cells all around the body. How big was the increase in infectivity? What are we talking about here?
Right. So we actually used a few different types of assays. So in the original work that we did, we used a very safe virus that we use in the lab, and we just decorated the virus or coated the virus with spike protein, either the original spike protein or the one carrying the mutation. And when we compared these two head to head, and we've engineered this virus to actually deliver a little fluorescent protein, so we can actually, the cells, when they get infected, they light up green. So it's really easy for us to precisely quantify which human cells are being infected. So when we initially did this with either the regular or the mutation, we found something between like a 2 and an 8-fold difference, and that's pretty big. If you say that you have a 8-fold greater chance of getting infected, that's a pretty serious change. Later, we actually were able to do pretty much the same experiment, except instead of using this other virus as kind of our main viral particle, we actually used the real deal SARS-CoV-2, which is the virus that causes COVID-19. And so we came up with this little trick where we were able to make SARS-CoV-2 particles that either have the regular spike or that have this spike that just carries this precise mutation, and again, test them head to head to see how well they infect cells. And of course, here, they don't make cells light up green. This is the real deal virus, so it actually causes the cells to rupture and die. And here, when we did this assay, we again saw an increase. It wasn't quite as high as what we saw with the pseudoviruses, not quite 8-fold, but it was still quite a significant increase, let's say around 2 or 3-fold. And so either way, whether you just look at spike kind of in isolation, or if you look at it in its natural context with the SARS-CoV-2 virus, in both cases, what we see is this tiny mutation, just one letter change, really increases the infectiousness of the virus.
Right, yeah. 8-fold is quite an increase. Now, this is not just a respiratory disease, it is another one of the implications here, but one that involves multiple systems. What's the knock-on implication for medicine, kind of knowing that we're not just dealing with something that causes acute respiratory infection, but something of greater significance?
Yeah, so we were surprised to see this and to hear this reported. Initially we did all of our work in a human lung cell line. So these are cells that are derived from human lung tissues. But we found that our results were really consistent in pretty much any human cell type we tried. So as I mentioned, we also looked at liver cells. We also looked at colon cells. And it looks like the virus is really able to infect any of these. And we were surprised that in some of the non-lung tissues, we didn't even have to boost levels of that ACE2 receptor. Some of them just naturally have high levels of ACE2, which is what spike binds to get into cells. And so I think the emerging medical evidence is really that this is going to be a multi-system, multi-organ disease. In fact, one of the best early diagnostics that people who have had COVID-19 report is that they lose their sense of smell. This seems to be one of the earliest things. And we know that the human olfactory system relies on neurons, olfactory neurons. And so what this is saying is that not only is this virus able to infect, whatever, lungs, liver, kidneys, but also the nervous system. And of course, this is something that I think merits further study. Of course, for most folks, they get their sense of smell back within weeks to months after they've had COVID. But it's very important for us to understand the long-term effects that can have on neurons. Neurons, unlike many other organs in the body, don't really regenerate that much. So it's I think there's going to be years of work that need to be done for us to fully understand the impacts of COVID.
Sure. And as you pointed out to me as we were talking right before this conversation, spike has a role in many of the vaccine development programs that are underway. How does this new understanding of the mutation affect the vaccine efforts?
Yeah, that's a great question. So there is approximately 150 different vaccine programs in different stages of clinical development, all the way from totally preclinical to phase 3 trials right now. And many of the ones that are most advanced, of course, had the earliest head start. They started early. And as I mentioned, the spike mutation only came up in February of 2020. Many of these, like for instance, the mRNA vaccine from Moderna, they started their trials in January. And this mRNA vaccine uses a full-length spike protein, and it uses, of course, the version without the mutation. Now, there's reason to believe that because that particular vaccine, for instance, encodes the full-length spike, it should be fine, because even though there's just one, this mutation just changes one piece of the protein, the rest of it can be used by our immune system to generate antibody-based protection or T-cell-based protection. But anytime you have a vaccine that doesn't perfectly align with the disease you're trying to treat, there's always a worry about how the mutation might impact immunity. And I think this is just something that needs to be tested. It'll very clearly be tested because, as I mentioned, now sitting here in July, virtually all the coronavirus that's currently active in the world carries this D614G mutation. So any patients being treated now almost certainly carry this mutation. The worry really is with some of the vaccines that have much, much smaller fragments of the spike protein. So some of them, whether they're DNA vaccines, RNA vaccines, or protein-based vaccines, can carry maybe much, much smaller pieces, like little peptides of the spike protein. And so the worry here is if those smaller pieces maybe just overlap this portion that has undergone this mutation, that there might be less immunity than you would get to the original virus. Beyond this particular mutation, I think the story serves as a little bit of a warning, perhaps, that we do have to be vigilant. This virus is just like any other organism on Earth, is capable of mutating and changing. And so one thing that I think is kind of a positive that comes out of this is we now have, with the efforts that have been put in to characterize the spike mutation, we kind of have a nice pipeline for any future mutations that come up to really very quickly try and understand how do they work, how do they change infectiousness, and how do we try and figure out the mechanisms behind that.
That's great, yeah. Okay, and so, this mutation, we know that it increases the viral load and increases infectivity. What we don't quite know yet is the impact on deaths and mortality. Is that something, that concerns you? I mean, obviously, not knowing it is a concern, but how much is that a concern, and what are the other worrying aspects of this mutation?
Yeah, so some of the other studies that have looked into this mutation have tried to analyze hospitalization rates or severity, and the data we have so far is a bit sparse, but what we do have is there has been observed by one group at least a higher incidence of ICU stays, which kind of is a surrogate for more severe COVID, with folks that have the D614G mutation. And so that's, I should say that trend, it was a trend, it's not statistically significant, but there was an enrichment with ICU stays. So right now, I don't think we have clear evidence that it increases severity of the disease, but it might be the case that the disease is more transmissible, it just gets around to more people with the same severity, or it might be the case that it does result in a slight increase in severity. Certainly, if you think about it from the perspective of individual cells in the body, if the virus is able to transmit more effectively, we often think of transmission between people, like, you're giving me COVID or I'm giving you COVID, something like that. But, within our bodies are communities of individual cells, right? And if they can transmit to each other more easily, that can also, that might also correlate with disease severity. But there's no clear data about that right now.
Mm-hmm, mm-hmm. And the the infection rate or the transmission rate at least in the New York area, I think is below 1, which they kind of use as another surrogate marker for kind of the whether we're a hotspot or not. It's good to see that is kind of being kept at a minimum. But perhaps, as we see this mutation making its rounds, I hope we'll keep an eye on that number. Just to kind of finish off the interview here with you, Dr. Sanjana, will we find a way to eventually stem the coronavirus? I guess that's a $64,000 question, but where do you see this going?
Yeah, I certainly hope so. So I think everybody has been shocked with really the effect of a global pandemic, unlike any that anyone alive today has ever seen before. And my hope is, of course, that the efforts, not just the efforts in my lab, but the efforts that really so many scientists around the world are making right now to try and contribute their knowledge, their abilities to come together and work on this problem will result in some accelerated therapeutics, some accelerated diagnostics, and of course, an accelerated vaccine. And I think the best indications that we have by folks who are very plugged into vaccine trials like those at NIH is that, there's never been, this kind of pace of development in biomedical research, period. And so just based on that, I'm also quite optimistic that within 6 months to a year, to maybe to a year and a half, we will have a widely available, reliable vaccine, and that will help end this pandemic. But, I think there's a lot that we can learn from this experience, and I hope that we're able to learn from it because we have the tools today with things like rapid gene sequencing to really be more vigilant, to have better surveillance, to understand when something has a pathogen emerges, and to really mobilize resources to that. And I think that those don't all have to be, we're talking about things like sequencing and CRISPR. Those are very high-tech things. They don't have to be so high-tech. I think a lot of us have seen over the last few months how effective things like wearing a mask and washing our hands are. But I think having that science-based response that comes in and can critically inform us will be very important moving forward. And so, it's probably not a unique thing to me, but I really do hope that we can learn from this and prevent it not just in our lifetime, but hopefully for future folks for a long, long time.
Absolutely. This is really a living lesson in pandemic preparedness, isn't it? Yeah. Okay, well, and that's reason for hope as well. So, I want to thank you, Dr. Sanjana, for joining us. This has been another fascinating discussion.
Thank you so much for having me.
Of course, and I want to remind our audience that you can join us virtually for live discussions on the future and networking at the MM&M Transform Conference. That's September 30th and October 1st. The link to register is in the description. Thanks for joining us, everybody. Take care.